Thank you everyone for attending this presentation. It's a pleasure to have you all here, and I especially appreciate the time and the dedication of the members of the jury. So today I'm going to present the results of the work that I have been doing during these last 5 years of my PhD, which has been focused on developing neuromodulation interventions and EEG biomarkers for executive function in healthy and clinical populations, as the title indicates. So the outline of the talk consists of an introduction in which I'll cover the basic concepts of executive functions and their anatomy, their assessment, these executive syndromes, and EEG. And then I'll go into the details of the studies that I have been working on. So as you will see, this thesis is divided in two parts. So the first one consists of two studies about TDCS neuromodulation of executive functions. And the second one also has two studies about deep learning applied to EEG for biomarker discovery. And finally, I'll discuss and summarize the conclusions, and I'll open the Q&A session. Let me change this to the laser. Okay. So let's start with introduction. So as some of you may know, executive functions is a broad concept that is used to describe the meta level of cognitive functions that are essential for successful adaptation to all kinds of environments, including, for example, professional environments and social environments. And through history, there have been several models of executive functions, but here we can see one of them proposed by Daphner and Searle in 2008, in which they divide executive functions into first order and second order. So the first order include the most essential functions, such as, for example, working memory, which is the ability to store and manipulate information for a brief period of time. Inhibition, which can be thought as the suppression of a cognitive action, such as resigning to an immediate reward in exchange for a better reward in the future. Initiation, which is the purposeful commencement of a task or a mental activity, and monitoring, which is the ability of keeping track of the environment with a particular expectation of how it should behave. And then second order executive functions include more complex functions that are built upon the basic ones, such as, for example, planning, organization, resisting interference, or affect regulation, etc. On the anatomical side, the prefrontal cortex is thought to be the central hub of executive function networks, together with its subunits, which include the paralimbic cortex, which is more in charge of initiation and monitoring, and it includes the anterior cingulate, the pearl factory, and the caudal orbitofrontal area. So it would be the orange part in this figure. And then the heteromodal association cortex, which is more in charge of working memory and inhibition, and it includes the dorsolateral prefrontal cortex, the temporal lobe, and the inferior parietal cortex. So it would be the green part in this figure here. And in this presentation, I'm going to focus mostly on the DLPFC, which is thought to be the area mostly involved in the executive functions that I have studied in this project. So executive functions are usually assessed in different ways. There are self-assessed questionnaires, there are also neuropsychological batteries of tests, and there are also behavioral or cognitive experimental tasks, such as the flanker or the stop signal task that we have used in this study. And in this task, usually subjects are presented with a series of stimuli, and they usually have to perform a specific response to each of them, while their performance is being evaluated in terms of reaction time, accuracy of responses, or even using physiological data like EEG or MRI, like we have done here. On the clinical side, there are a number of clinical populations with impaired executive functions, which include, for example, neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, or Huntington's disease, infections of the central nervous system like neurosyphilis, traumatic brain injury, diseases affecting the white matter tracts like multiple sclerosis, neuropsychiatric conditions like schizophrenia, bipolar disorder, depression, among many others, and also neurodevelopmental disorders like autism or ADHD. And in this talk, I'm going to focus mostly on ADHD and Parkinson's disease, which are the two clinical populations that I have studied. On the physiological side, EEG or electroencephalography is the measurement of the electrical signals from the brain, and it constitutes a direct measure of neuronal function. And there are different ways to analyze EEG signals, mostly in the time domain and in the frequency domain. So in the time domain, one of the advantages of EEG is that it has a high temporal resolution that offers a great opportunity to further understand the physiology of executive functions. And more specifically, event-related potentials are specific patterns of EEG signals that are time-locked to specific external stimuli events, and they have been established as relevant biomarkers of executive function. So in this figure here, we can see an example of an ERP pattern that can be detected after a stimulus is shown to a person, like an image or a sound. And here we can see different examples of ERP components. So P200, for example, is this positive peak here, which is associated with selective attention. And 200 is more related to the attentional effort that is needed to perform a specific task. And P300 is more related to the conflict resolution or the cognitive control or inhibitory control. And in the frequency domain, EEG signals are analyzed as periodic signals that oscillate at different frequencies, and each of them is associated with different cognitive processes, such as, for example, the delta waves that appear during REM sleep, the alpha waves that appear during periods of relaxation, etc. And there are different ways to represent the EEG signals in the frequency domain, mostly using either the spectrum of the signal, which represents the power in each frequency band, or the spectrum of the signal, which is a representation of the power at each frequency and how it evolves over time. And you will later see that in this thesis, I have used the ERPs in the time domain, to assess the effects of neuromodulation on executive functions, and the spectrum in the frequency domain to find disease biomarkers using machine learning. So moving on to the actual studies, as I mentioned before, this thesis is divided in two parts, and each part includes two studies. So the first part is entitled TDCS Neuromodulation of Executive Functions, and it is focused on using EEG to assess the cognitive and the physiological effects of TDCS on executive functions in healthy and ADHD populations. And these are the two studies included in this part. Then the second part is about using deep learning for biomarker discovery, and it is focused on exploring a deep learning approach to classify and characterize different clinical populations on the basis of their EEG data. So let's move on to the first part, in which I'll cover the concepts of ADHD and neuromodulation, and then I'll go through the aims, the methods, the results, and the discussion. So in this part, I'm going to focus mostly on attention deficit hyperactivity disorder, which we know that affects around 5% of the population, although there's a wide variability between children, adolescents, and adults. And we know that it is a signature disorder of executive functions characterized by deficits both in high-order cognition as well as in response to rewards, such as the well-known ability to delay gratification, for example. It has also been shown that the DLPFC is related to the executive functions that are mostly impaired in ADHD, such as selective attention, inhibition, and processing control. And the problem is that pharmacotherapies exist, but many patients do not experience complete relief in the long term, or they have side effects. And there are some studies that have shown promising results in the enhancement of executive functions in ADHD using what is called neuromodulation techniques. And before going into more details, I would like to introduce you to the concept of neuromodulation, which is formally defined as the alteration of nerve activity through the delivery of a stimulus, such as, for example, electrical stimulation like we do here, or chemical agents, to specific neurological sites in the body. And there's quite a lot of evidence of the efficacy of neuromodulation techniques in both healthy controls and clinical populations like Alzheimer's disease, Parkinson's disease, stroke, schizophrenia, depression, among many others. And here I would like to show you a video as an example of the effect of one of these neuromodulation techniques on a patient that has Parkinson's disease. So in this case, they are using an implantable technique, which is called deep brain stimulation, in which they implant stimulation electrodes inside the brain to induce the current in the target areas. So let's see if it works. So here we can see the patient who has a strong tremor caused by the actual disease. And when they turn on the stimulation, this is the device controlling the stimulation, we will see that the tremor starts to slowly decrease until it completely disappears, like this. And then we will see that he can perform some normal tasks, like he can speak normally, He can even do some motor tasks like finger tipping. So he can perform almost all his daily life activities without any problem. And these are amazing results, and there are also some studies assessing the effects of other neuromodulation techniques in ADHD, but most of them only assess the behavioral part or the behavioral effects, but not the actual physiological effects like using EEG data, like we have done in this study. So in this thesis, I have explored a particular type of neuromodulation technique, which is called transcranial current stimulation, as a way to enhance executive functions in populations like ADHD. And specifically, the broad concept of transcranial current stimulation involves three main types of stimulation. So direct current, which is a constant signal like this one, alternating current, which is a signal with the shape of a sinusoid at a specific frequency, and random noise, which is actually a combination of sinusoidal signals at different frequencies that just ends up being random noise in all the frequency band. And these three different types of signals are used for different purposes in research. So, for example, TDCS is used to increase or decrease the excitability of neurons in a constant manner. TDCS is used for the entrainment of specific rhythms or frequencies in the brain. But in this case, I'm just going to focus on transcranial direct current stimulation, or TDCS, which is the one that I've analyzed in this study and also the one that is mostly used in research. So the way in which TDCS works is that it introduces small doses of current into the brain that create weak electric fields that change the membrane potential in cortical neurons. And this in turn changes the probability of the neurons to fire. So according to this, there are two main types of stimulation. A nodal stimulation that occurs when the electric field enters the cortex, which causes a depolarization of the membrane, and this creates an excitatory effect, so the neurons increase their firing. And cathodal stimulation, which occurs when the electric field points out of the cortex, which causes a hyperpolarization of the membrane that creates an inhibitory effect, and the neurons decrease their firing. And then as a secondary effect, at the neurotransmitter level, we know that TCS or TCS also affects the balance between excitation and inhibition by modulating the GABA and the glutamate concentrations. So the standard TTCS montages consist usually of two electrodes applied to the scalp from a current-controlled battery like this one, with the current flowing from the anode electrode to the cathode electrode. So the anode is defined as the electrode that injects the current into the brain, and the cathode the one that extracts the current out of the brain. And fortunately, the current TDCS devices like the one that we used in this study, they offer more complex approaches that have multi-channel montages, for example, with more than one anode and one cathode, with smaller surface areas. And this allows for improved focality and also the possibility to stimulate multiple areas or multiple networks simultaneously. So the specific goal of these two studies were to, first of all, to assess the effects of TDCS on executive functions, both in healthy subjects and ADHD patients. And specifically, we wanted to investigate the role of the laterality of the DLPFC to further understand whether the left or the right DLPFC play a more significant role on executive functions. And second, we wanted to find EEG physiological biomarkers of executive functions to try to shed some light into the mechanisms of action of TDCS, and also to assess the state-dependent dynamics of this technology, which I will go into more detail later. So the study design for this part consisted on 20 healthy controls and 20 ADHD patients who performed 3 sessions. And for each session, they first performed 2 experimental tasks while we measured behavioral and EEG metrics related to these executive functions. Then they received 30 minutes of stimulation at 2 milliamps targeting the right DLPFC, the left DLPFC, or sham, which is like the placebo of stimulation. And here we can see the modeling of the electric field of the montages that we use to stimulate the left DLPFC and the right DLPFC, which has consisted on a simple bipolar montage with the nodal electrode over the left or the right DLPFC and the cathodal electrode on the contralateral supraorbital area. So just a simple bipolar montage that is used in these studies. And then they perform again the behavioral task while we measured EEG and behavioral metrics again, so that we could compare before and after stimulation effects. And just to give you some more details about the task, they perform two tasks, the flanker task and the stop signal task. So the flanker task is a cognitive task that measures attention and inhibitory control, in which subjects are presented with a certain stimulus. In this case, it's an arrow like this one's, that is surrounded by distracting arrows that can point to the same or to the opposite direction as the central one. So the goal for the subject here is to press the left or the right arrow keys depending on the direction of the central arrow and just trying to ignore the distracting ones. And according to this, there are two types of trials. So the trials that have the flanker arrows pointing to the opposite direction as the central one are called incongruent trials, as it is more difficult for the subjects to press the correct response. And the trials in which all the arrows are pointing to the same direction are called congruent trials, as they do not require additional effort to ignore the distracting ones. On the other hand, the stop signal task measures inhibition and impulsivity, and there are also two types of trials. So the go trials, in which the subject is presented with letter A or letter Z, and they have to press the left arrow when the letter A is presented, and the right arrow when the letter Z is presented. However, some of these trials will be followed by a stop signal, which indicates that the participant has to withhold the response, so they do not have to press any key. And these trials are called stop trials or no-go trials, and they are the ones that measure the inhibition capabilities of the subject because they force them to cancel a response that has already been initiated internally. And just to give you an idea or a very basic idea of the process that I followed to extract the ERPs, it consists on these steps. So first we clean the raw EEG signal from external interferences and motion artifacts like eye blinks like these ones or muscle movements. Then we filter the signal usually between 0 or between 1 and 20 hertz, which is the frequency range where these components are located. Then we cut the signals into different trials, taking as a reference the markers that are located at the onset of each stimulus. And finally, we average all those pieces of signals to obtain the final ERP waveform, which is much cleaner than the original single trials because all the noise has been removed by the averaging process. So these are the behavioral results that we obtain for the flanker task for healthy controls and for ADHD patients. And these bars here are showing the reaction time before and after stimulation, targeting the left DLPFC, right DLPFC, and sham. So what we found here is that applying TDCS to the left DLPFC makes the participants faster, or the reaction time decreases, when replying to incongruent trials for both healthy controls and ADHDs. And it is worth to mention that there were no significant changes in accuracy for better or for worse, but there is still no trade-off between the effects of TDCS on reaction time and accuracy. So that means that reaction time is improved, but not at the expense of worsening the accuracy. And the nice thing about these changes on the behavioral side is that they were correlated with some changes on the physiological side, as you will see in the next slide. So these are the ERP patterns that we obtain before and after stimulation for healthies and for ADHD patients again. And here we can see that the improvement in reaction time after left stimulation is correlated with an increase in the amplitude of P300, which is this positive peak here, and a decrease in the amplitude of N200, which is this negative peak here. So according to the literature, an increase in P300 is associated with an improvement in conflict post-processing and inhibitory control, while a decrease in N200 is related with an improvement in the efficiency of the attentional resources that are needed to perform the task. So we interpret this improvement in reaction time as an improvement in these two executive functions as shown by these physiological changes. And there was a slight difference between healthy controls and ADHDs that we probably cannot see here, but it is that the decrease in the amplitude of N200 was only significant for healthy controls, but not for ADHDs. And this is normal and this is expected, as there are many factors that may affect the efficacy of TDCS, such as the higher heterogeneity in the signals of the ADHD patients, the severity of the symptoms, or the cortical regions involved in the pathophysiology of the disease, etc. And additionally, note that the distribution of the power in these scalp maps show that the largest changes in these ERPs occurred mostly around the area of the anodal electrode, which means that it's matching the laterality of the stimulation. So if we stimulate on the left DLPFC, the largest changes occur around the left DLPFC, and the same for the right, and nothing for sham. So in the case of the stop signal task, we also found a significant increase in reaction time in GO trials after stimulating the left DLPFC, which is again correlated with an increase in the amplitude of P200, which is this positive peak here. And in this case, this is associated with an improvement in visual selective attention according to the literature. And although intuitively this may seem like a bad result because it's like the patients are getting slower in their responses, it's actually considered a good effect if we put it in the context of the stop signal task. So we need to think what the patients are trying to do here. So in the case of the GO trials, what they are trying to do is they are trying to wait to see if the stop signal appears after the GO trial. So the longer they wait, the less impulsive they are. So in this case, we could consider this increase in reaction time as an improvement in visual selective attention, which translates into less impulsivity and better proactive inhibition. However, in the case of the stop trials, there were no significant changes in the stop signal reaction time, which is defined at the time as the time takes for the subjects to internally cancel the response that has already been initiated when they see the stop signal, which means that there is no improvement in reactive inhibition or action cancellation. So it is important here to note the difference between proactive inhibition and reactive inhibition. So proactive inhibition is the act of waiting for the stop signal to appear in a proactive manner, and reactive inhibition means canceling a response that has already been initiated internally. And in order to confirm the value of these ERPs as cross-sectional biomarkers, and of course also to support the interpretation of these results, we assessed whether the amplitude of these ERPs was correlated cross-sectionally with the behavioral performance. So indeed, we found that the amplitudes of P200, N200, and P300 were significantly correlated with reaction time in a cross-sectional trial-by-trial basis. So for example, the greater the P200 and the P300 amplitudes, the faster the reaction time, and the smaller the N200 amplitude, the faster the reaction time. So this basically provides empirical evidence that supports their value as cross-sectional biomarkers of cognitive performance. And finally, we also assess the state-dependent dynamics of TDCS. So the concept of state dependence refers to the idea that the effect of an intervention like TDCS depends not only on the stimulation parameters, but also on the initial state of the patient, both physiologically and behaviorally. And specifically for these studies, we found that those subjects with more impaired executive functions at baseline before the stimulation, they were the ones who had a greater improvement after a stimulation. So for example, the smaller the P300 at baseline, the greater is the increase in P300 after stimulation. So the greater is the improvement after TDCS. And this confirms the idea that TDCS has a state-dependent nature, which means that it has a greater effect on physiology and behavior in those subjects with baseline physiological signatures that are indicative of less adaptive processing. So these results have important implications for treatment development because they suggest that beyond the stimulation parameters, clinical studies may also benefit from controlling the patient's state before, during, or after stimulation in order to minimize response variability and also to maximize the therapeutic effects of TDCS. So as a summary of our findings for the first part, we found that anodal TDCS targeting the left DLPFC leads to behavioral improvements for both healthy controls and ADHD patients, which is correlated with an improvement in the physiological ERPs related with attentional efficiency and cognitive control in the case of the flanker task, and proactive inhibition or impulsivity in the case of the stop signal task. However, we did not find significant changes in the stop signal reaction time in the case of the stop signal task, which suggests that TDCS did not improve reactive inhibition in this case, And this supports the existence of different impulsivity subconstructs with overlapping but distinct anatomical substrates. In addition, we also confirm the amplitude of these ERPs is correlated with reaction time on a trial-by-trial basis, which provides empirical evidence that supports their value as biomarkers of cognitive performance. And finally, we also showed that the baseline physiology of the patient before stimulation determines the cognitive and the physiological effects of DBCS, which confirms the state-dependent nature of DBCS. So I'll review again these conclusions at the end of the presentation, but for now, let's just move on to the second part, in which I'll cover the concepts of machine learning and deep learning, and then I'll go through the aims, the methods, the results, and the discussion. So first, I would like to introduce you to the concept of machine learning and deep learning, which I'm sure that you've heard about as they have become increasingly well known in many different areas. So machine learning can be described as the concept of training a system to make a classification or a prediction by using large amounts of data. So for example, training a system to predict the evolution of the prices of a house, or depending on their characteristics and the historical data, for example. And more specifically, deep learning is a more complex subset of machine learning algorithms that use what is called deep neural networks, which can detect more complex patterns in the data and can also provide higher levels of accuracy in many problems. So here an example could be training a camera in a car to try to detect the road and the obstacles in front of the car so that it can drive autonomously eventually. And just out of curiosity, deep neural networks are inspired by the actual architecture of biological neurons, in which the connections between the neurons are modeled as weights, and the output of each neuron is like a linear combination of the inputs multiplied by the corresponding weight. So similar to the idea of the synaptic neurotransmission between neurons. And in this thesis, I have used a specific type of deep neural network called convolutional neural network, or CNN for short, which was originally designed in analogy with a primate visual system with the goal of classifying images. So, for example, they are widely used in applications like facial recognition or autonomous vehicles. And for the sake of time, I won't go into much details about how they work, but basically they extract different patterns called feature maps from the input image that are later used to identify the most relevant parts for the image for the classification. So for example, if we want to train a system to classify between apples and oranges, the system will just learn the key differences between them. So like, for example, the color, the shape, etc. And in this thesis, I have applied a CNN to EEG data in order to classify ADHD patients from healthy controls and also to predict whether a patient will develop Parkinson's disease. So the goals for the first study of this second part is to find an EEG biomarker that discriminates 20 ADHD patients from 20 healthy controls by using a deep learning approach that can find specific patterns in their ERP data that characterize each group. And then in the second study, we have an EEG data set of 90 healthy controls and 120 patients with REM behavioral disorder, or RBD. And this is considered a prodromal stage of neurodegenerative disorders like Parkinson's disease or dementia with Lewy bodies. Then some years later, some of these patients were eventually diagnosed either with Parkinson's or with dementia with Lewy bodies, while the rest remain idiopathic RBD. And our goal here is to use a similar deep learning approach as the one that we used in the first study to see if we can find any patterns in the EEG that allow us to predict what patients will eventually develop Parkinson's and which ones will just remain idiopathic RBD patients. So here we can see in more detail the process that we followed. So for the first study, we used the ERP data that we recorded while the healthy controls and the ADHDs were doing the flanker task. And for the second study, we used resting state EEG data from this data set of patients with REM behavioral disorder. Then we processed the data in order to clean all the EEG from external interferences and movement artifacts. Then we computed the spectrum of the signal for each channel. And finally, we used these images or these spectrums as the input into a convolutional neural network that was trained and cross-validated using a live per out approach, which I will not explain with much detail, but for those of you who are interested, It basically consists on splitting the data between different training and test sets and repeating the process over and over again several times to calculate the average performance of the network. So this basically ensures that the network doesn't overfit and maximizes the generalizability of the network to other types of data that have not been used as in the training process. And for those of you who are curious about the technicalities of the network, this is the architecture that we use for the CNN, which consists of 2 convolutional layers for feature extraction and 2 linear layers for the classification. And for comparison purposes, we compared the performance of the CNN with 2 other state-of-the-art networks, in this case, a recurrent neural network, which is also a deep neural network, very similar to the CNN, so we expect them to be quite similar in terms of performance. And then with a shallow neural network, which is just a very basic type of network that we use just for comparison purposes. And I'm not going to explain with much details for the sake of time, the architectures, but I'm happy to discuss them later if anyone is interested. So once a network is trained, we used what is called a feature visualization technique called Deep Dream, which was designed by Google a few years ago, and it basically creates artificial images representing the most relevant features for each class. So, for example, if a network has been trained to classify between dogs, flowers, and buildings, it will create artificial images like this by exaggerating the features that it has been trained to look for in each class. And this will be helpful for us to understand what patterns in the EEG data are the most relevant ones for the system to differentiate between patients and healthies. So for the first study, we found that the CNN trained to classify ADHDs from healthies reached an accuracy of 88%, which means that it was able to discriminate ADHD patients from healthy population 80% of the time, or 88% of the subjects. Then the RNN performance was very similar as expected, around 86%, and it clearly outperformed the shallow neural network that reached around 78%. Then we also compared the performance of the ERP spectrums with normal resting state EEG data, and we found that the ERP spectrums provided better performance for all the networks. And this is probably because, unlike the resting state EEG data, the ERPs engage those executive functions that are impaired in ADHD. Finally, we also looked at the performance of each single EEG channel and found that the frontal and the parietal channels were the ones providing better performance, probably because they are the ones that are less contaminated by motion artifacts like eye blinks or muscle movements. And also note that the performance of the electrodes located at the left hemisphere, like F3 and P3, is lower compared to those located in the right hemisphere, like F4 and P4. So this suggests that there may be some interhemispheric differences in the features driving the classification that should be further analyzed. So here we can see the results of the Deep Dream technique, which generated an image representing the spectrum of the healthies and the spectrum of the ADHDs. So if we compute the difference between the two, we obtain this other image here, and this spectrum shows that the main difference between healthies and ADHDs is, first of all, an increased power for the ADHD group in the delta theta band, between 3 and 6 hertz, and a decreased power in the alpha band between 7 and 12 hertz along the entire ERP. And according to the literature, these two changes have been associated with attentional and inhibition deficits, which is consistent with the ADHD impairments in this population. So what this is doing is this is actually validating the system because it proves that the network is not detecting noise, but it's detecting an actual biomarker that has been validated before in the literature. Then for the second study, we applied the same methods and the same architectures, and we obtained the following results. So first, we could predict what patients would eventually develop Parkinson's disease as compared to healthy controls with 80% accuracy up to 6 years in advance before they develop any symptoms. Then we could also predict what patients eventually develop Parkinson's disease or dementia with Lewy bodies with 72% accuracy as compared to those who would just remain by the Deep Dream technique, and we found similar features. So we also found a decrease in the alpha power around 10 hertz, and an increase in the theta power, around 6 hertz. So this is consistent again with other studies that found similar alterations in the EEG of Parkinson's patients, which may be related again to their deficits in working memory, planning, and visual attention. So again, this provides us with evidence that the system is driven by actual physiological changes and not just by noise or any other artifact. So as a summary of our findings for the second part, we could diagnose ADHD patients from healthy controls with 88% accuracy, and we could also predict who would develop Parkinson's disease with 80% accuracy up to 6 years in advance. We consider this system to be particularly interesting because it basically mitigates the need for EEG processing and feature selection, because these deep neural networks, basically, they learn the features that are important for the classification or the prediction without you having to tell them specifically where to look at in the image. And through the use of the deep dream technique, we identified what features drive the classification, and this provides a mechanistic model to further understand the pathophysiology of the disease, and this can eventually transform these features into biomarkers to transform or to diagnose or predict the disease. So as a summary of the conclusions for both parts, in the first part, we found that a nodal DCS over the left DLP of C using a simple bipolar